Students, microscope is indispensable to a biologist. As you enter into a lab, you have to be expert. You should know how to deal with a microscope. Let us learn the parts today. This is a compound microscope. All of you might have seen it, correct? So, let us look at the parts intricately today. The base of the microscope is not solid. You can see the bifurcated nature. Why is it so? And there are some rubber pads below. This is just to stabilize the microscope, so that wherever you are putting it, it might not topple down. And the center of gravity is also kept low. If your base was solid, it could have toppled. Next, we move upwards. This is the foot. This is the foot of the microscope. Here you will find there is a screw. This screw is known as inclination screw. You can use this screw to tilt your microscope the way you want to watch it. That is why it gives you an extra advantage. You can sit and look into your microscope depending upon your requirement. This part is known as the arm. This part is bent. Arm is not straight, it is bent to get a good grip, of the microscope. This part of the microscope is known as the body tube. This part is the body tube and this part is the nose piece. So, what did we learn? The mechanical parts comprises of the base, then we have the foot, this is the arm, this is the body tube and this is the nose piece. And there are two more screws apart from the inclination screws. What are they? This is the coarse adjustment screw and this is the fine adjustment screw. These screws help you to get to see and identify your specimens very closely and intricately. We will come to it later on. In between these two, we find there is a stage kind of thing. This is the stage where we put in the slide to have a clear view of the specimens, correct. Now, coming back to the optical parts of a microscope, there are few lenses. This lens that you see in the nose piece, usually it is of 10x. It is of 10x. What is 10x? It will give you a magnifying power 10 times the size of the object. This is 10x eyepiece. Below this body tube, you find there is a rotating structure. It will be prominent to you. You can see there are 3 lenses posted here. 3 lenses are posted here. These lenses can be removed. If you do not want, you can remove it from here also. And the nature of this circular disc is also revolving. What are these types of lenses called? These lenses are known as objective lenses. Now, these lenses are of 10x, usually they are 10x and 45x. One is used as a low power, another one is used, 45x is usually used as a high power objective lens. And the third one is used specially on special occasions. I have one here which is 100x. So, when I am using suppose 10x here and I am watching it under 10x here. So, how many times is the specimen being magnified? 10 into 10, that means 100 times. You are getting the diagram or the picture. If you want higher magnification power, then we go in for oil immersion lens, where you, like for example, you want to see the inside material of a cell. At that time, what do? The permanent slides that we have or the mounted slides that you have, there is a cover slip on top of it. On this cover slip, we just put in eucalyptus oil or olive oil and then we put it on the stage. We put it on the stage and we position it under 100x. When we put it under 100x, then it enables us to have a much closer view of the specimen. This is only done for certain special slides, not usually. Usually we position the specimen under low power and then we transfer it under high power to have a good observation, correct. One more hidden thing is there in the microscope, we cannot ignore it. What is that? I have to tilt it. Can you see there is a mirror? This mirror is the reflecting mirror. It is plane on one side and it is concave on the other side. It is plane on one side and concave on the other side. What do we call it? It is a reflecting mirror. Okay. And just on top of that, you find there is one iris diaphragm. In our eyes we have iris. What is the work done here? This diaphragm is perforated. As we shift it, it changes the aperture inside. The purpose of having this mirror and the diaphragm is that we focus the light rays. So that it is concentrated here through this condenser. This part is the condenser lens. In this microscope, remember, the purpose of this lens is to condense the light, whereas the purpose of your objective lens and ocular lens is to magnify the image. These two are for magnification, but here this condenser is for concentrating the light rays, so that maximum amount of light can enter. Besides this, we have extra screws in some of the compound microscope. This enables us to shift when we are placing a slide here and keeping our eyes here. We are not going to remove our eyes on and off. With the help of these screws, you can change the position of the specimen to find out a specific structure as per your need. It can shift it towards your left or towards your right as required. When we know all the structures, we have to be very careful how to handle our microscope. Sometimes on this stage glycerin people drop off, or some people drop off safranin, which has to be regularly cleaned, and the lenses have to be cleaned with a sterilized cotton or cloth with the help of xylene. Otherwise, sometimes bacterial or fungal growth is observed, which does not give us a clear vision. So, these are the precautionary measures which we have to keep in our mind. That is all. Thank you. Yes, students. So, as we have known all the components of a microscope, just a synopsis, let us summarize it, see how far you have registered. So, the parts of the compound microscope, we can put it under two headings. One is mechanical part, another one is optical part. The mechanical part comprises of your nose piece, the body tube or the draw tube. We have a stage which has the diaphragm below that. Then we have the bent arm and the bifurcated base. Whereas the optical part comprises of your ocular lens, which is usually 10x or sometimes 20, and the objective lens are of three varieties: low power, high power, and one oil immersion lens, which is usually of 100x. Next to that we have condenser. Condenser's purpose is very special. It is only for concentrating the light rays, and reflecting mirror is plane on one side and concave on the other to focus the light rays to the condenser. And that is all. Have a good day.